Hello everyone, and broken down into 4 sections, this is our biology project by New, Plawan and Win. Today we will be talking about DNA, proteins, mutations, and karyotypes. There are 2 parts. The first is the explanation of terminologies or concepts associated with each topic, and then the synthesis part, which will be in forms of mnemonics, analogies, cause and effect relationships, or real-life examples. Starting off with the topic of DNA, structurally speaking, we have the pentose sugar, or 5-carbon sugar, in the middle, a phosphate group attached to the 5' carbon, and a nitrogenous base attached to the 1' carbon. DNA is basically made from chains of what is called nucleotides, which were the 3 structures previously mentioned. What is a nitrogenous base? Well, let's take a closer look. It has 2 main categories: purines and pyrimidines. The main way to differentiate between these 2 types is that purines will have 2 rings, while pyrimidines will have 1 ring. In DNA, adenine and guanine are purines, and thymine and cytosine are the pyrimidines. In RNA, however, uracil replaces thymine. The internet is sometimes helpful, a mnemonic that can be used to remember these 2 terms, and the chemical basis associated with them is: purines are pure as gold. The first letter in as is A and represents adenine. The first letter in gold is G and represents guanine. The word pure is used to remember that adenine and guanine are both purines. And for pyrimidines, think of them like pyramids. Pyramids are triangular shaped, and think of these triangular shapes as sharp because they cut. C representing cytosine, U representing uracil, and T representing thymine. Now that we've gone over what these nitrogenous bases are, surely we have to ask ourselves the question, well, how do they pair with each other? Do purines pair with purines and pyrimidines with pyrimidines? Well, there are certain rules, as we call it, base pairing rules, essentially rules that govern how nitrogenous bases should pair. Now, ideally, adenine pairs with thymine and uracil in RNA, and guanine pairs with cytosine. There's a pattern here. Purines tend to pair with pyrimidines, and pyrimidines with purines. Why? Well, as we recall, purines have 2 rings, whereas pyrimidines have 1 ring. If a purine bonds with a pyrimidine, the space between the strands is taken up by 3 rings. If a purine with another purine, that'll be 4 rings. And a pyrimidine with a pyrimidine, that'll be 2 rings. The space between the DNA strands cannot have this much variability. If the space between the DNA strands had 3 rings, then 2, then 3, then 4, then 3 again, that would be a notable change in the structure of DNA. All of the bonds made by nitrogenous bases are hydrogen bonds. We've been talking about a lot of these 2 strands of DNA. And yes, they have names. There is the sense strand and the antisense strand. The sense strand is the strand that is used in the transcription process, which is covered later. The antisense strand is complementary to the sense strand. These 2 strands are anti-parallel. After covering everything about DNA, what's the importance of it? Well, in a nutshell, DNA contains the instructions or recipes essential for growth and survival. Think of it like a cookbook filled with all the instructions on how to make these things called proteins now instead of words. Our body has its own language, which is contained in the sequence of the DNA sequence or the sequence of the nitrogenous base, specifically a 3-letter sequence. Right now we're going to gloss over the central dogma of biology, how DNA moves to RNA, which they turn into protein. Starting off with transcription, the DNA sequence is transcribed by RNA polymerase to mRNA, messenger RNA. This transcription starts with the RNA polymerase, reaches a 3-letter sequence, also referred as a codon. That starts the transcription process. The process ends when the RNA polymerase reaches another unique 3-letter sequence, instructing it to stop transcribing. The names of these unique 3-letter sequences that start or stop the transcription are referred to as the promoter and terminator respectively. Picture the phrase of transcription as the DNA sequence of being lectures from a professor. The RNA polymerase as the students ready to learn, messenger RNA as a blank piece of paper waiting to be written on. Now, of course, students don't write down every single word they hear or see in a lecture. There is junk DNA, or certain things that are not relevant. Student needs certain cues to know when to start taking notes. The promoter in this case is the professor stressing that a specific piece of information that they are talking about is important. That's when the RNA polymerase or the student start transcription or taking notes on the messenger RNA, also known as their notebook. After the transcription, there is a process called translation, which entails the creation of amino acids and eventually protein synthesis in what is called ribosomes. Taking some of the terminologies from transcription, imagine the mRNA, as the students note, being read by ribosomes, group of many students creating proteins or meaningful information, writing essays or even book based off the notes taken in class. Proteins. The word protein and amino acids has been used a lot, somewhat interchangeably. This section covers amino acids, how they fold and bond, first and foremost. Proteins are chains of amino acids. For proteins to be made, amino acids have to join together and fold in a specific way to create specific proteins. If you recall, a codon is a 3-letter sequence that corresponds to an amino acid. There are 20 amino acids in total, with their properties either being nonpolar, polar, acidic, or basic. When amino acids are folding, these are the list of bonds that are the strongest to weakest. A cysteine and cysteine bond, or a disulfide breed. An acid with a base, negatively charged with positively charged. Lastly, polar to polar bonds. Mutations. A simple definition of a mutation is the adding, removing, replacing, or altering of at least one part of the DNA sequence. This can be accomplished in many ways, from ionizing radiation to viruses to mutagenic chemicals to DNA replication errors. Well, what are the effects of these to the human body in real life? Well, first, ionizing radiation. Electromagnetic radiation, with just the right wavelength, can break DNA. Some examples of ionizing radiation is X-rays and UV rays from the sun. There's no need to get worried. We aren't exposed to X-rays every single day, and if you do go to the doctor, you'll notice that you will get to wear these vests. Well, they protect you from unnecessary exposure to radiation in parts of your body that the X-ray isn't scanning. In short, viruses cause mutations by making your body produce more, viruses, and this is usually done by just replicating the cell's nucleus with its own DNA, which is instructions on how to make more viruses. Mutagenic chemicals cause mutations directly affecting the base pairs, some achieved by inserting itself between base pairs, others by changing the structure of the bases. Now the real effect comes when the DNA is being replicated and those aforementioned changes are permanently implemented, adding or deleting a base pair permanently. DNA replication errors is quite broad. As previously described, things like viruses and mutagens can initially cause small mutations. When the DNA is replicating, these mutations get passed on to other cells, and in some cases, to entire generations, amplifying the effects of the mutation. This is also seen as DNA polymerase enzymes making a mistake, and instead of adenine, it adds cytosine instead. Karyotypes.
A big word, but if broken down to its roots, karyo meaning nucleus, and types in this instance meaning description, all it means is that it is a method of organizing chromosomes from longest to shortest, with the sex chromosomes last. The purpose of it? This is usually done to check if the person has the normal amount of chromosomes and doesn't have any syndromes or medical issues. How is it written? A typical karyotype of a male would look like something like this. 46, the normal amount of chromosomes, with 22 of them being homologous autosomes, chromosomes that don't determine sex, and the other 2 being sex chromosomes, comma, and XY, the sex chromosomes present in males. Why are karyotypes such a big deal anyways? Imagine yourself as a mother of an expecting child. You order a karyotype test intended to determine the sex of your child. When the test comes back, there are these words written in bold. 47, XX plus 13. A genetic counselor sits down with you and your husband to have a serious talk. The good news first. Your child is a girl. Now the bad news. Your child has an entire copy of chromosome 13, hence the plus 13 at the end, which makes the total count of your chromosome of your baby to be 47. An addition in part or whole of chromosome 13 is called Down syndrome. You are thankful to receive the information early, but are devastated by the misfortune.